Wonderful. I think we'll get started now. I want to thank everybody for being here today and so much for tuning in. We're pleased to have you join us for the California Current Ocean Acidification Network Roundtable discussion for December 2022. The title of today's presentation is High Partial CO2 and the Thermal Plasticity of Developing Pacific Herring Embryos During a Marine Heat Wave. This series is hosted by the California Current Ocean Acidification Network. In short, the series is intended to create a direct dialogue among industry members, tribes, natural resource managers, and scientists within the California Current ecosystem. With awareness of and access to the research and its applications and uses, the participants will be able to collaborate to better understand and to adapt to ocean acidification moving forward. There we go. We can go to the next slide. I'm Terry King, and from Washington Sea Grant, and a member of the California Current Acidification Network, and I'll be moderating today's presentation and running the logistics behind the scenes. Chris Murray is our presenter this afternoon. During the presentation, attendees will be in listen-only mode. You're welcome to type questions related to technical issues or questions for the presenter in the chat box at the bottom of your control panel. I will be monitoring those and the incoming questions, and we'll respond to them. And we'll hold we will hold them for direct questioning of Chris when we're done. We're also recording this session, and we'll share the recording on the California Current Acidification Network website. So Dr. Chris Murray is an experimental biologist who combines laboratory studies with field-based observations to better understand the processes that mediate phenotypic plasticity and flexibility under climate stressors like ocean warming, acidification, and deoxygenation. His work primarily focuses on fish early life history stages, of which constitute a critical window of sensitivity to environmental stress. Chris conducted his dissertation research at the University of Connecticut's Department of Marine Sciences before relocating to the Pacific Northwest, where he worked as a research associate with the Washington Ocean Acidification Center and the University of Washington. Currently, Chris is a National Science Foundation postdoc, investigator at Woods Hole Oceanographic Institute, where he's studying the multi-generational impacts of coastal hypoxia and acidification in a key forage fish species. Now I'd like to turn this over to Chris for your presentation today.
Awesome. Thank you so much, Terry. And it's great to have everybody here. Just a quick summary of the talk today. I'm going to talk a little bit about a paper that we recently published in the Journal of Experimental Biology that details some experimental work where we evaluated the influence of acidification on the thermal plasticity of Pacific herring embryos. From there, I want to quickly summarize a follow-up study that I did with some collaborators at Western Washington University, looking at some of the more finer questions that were actually turned up during this initial study, and then some work looking at acidification and disease in Pacific herring as well. So a quick summary or an overview of my research motivations. I'm primarily motivated by how organisms are adapted to the natural variability of their environment, and with the particular context of emerging climate change stressors. So the big three of warming, acidification, and deoxygenation. But more recently, I've become interested in the idea of extreme events, punctuated moments of temporal variability that kind of fall far outside the normal range of variability that organisms typically encounter. And some good examples of this are things like marine heat waves, which folks in the Pacific Northwest have become quite acquainted with, but also more marine specific scenarios like acute hypoxia and acidification that occur over the temporal duration of hours to days. So my work while I was a postdoc at UW and now primarily focus on what are the limits of environmental tolerance during these acute extreme events, and what might be the mechanisms of phenotypic plasticity that allow organisms to withstand these emerging threats. So just to talk about this a little bit schematically, the panel on the left shows typical environmental variability over the course of either a day or an hour or a season You might recognize this as a temperature fluctuation across the four seasons. This sine wave could also be oxygen fluctuations in a nearshore habitat over the course of a day-night or a tidal cycle. And generally, it's much more noisy and irregular than these clean sine waves. But the general pattern is there. And organisms that live in these environments are well used to or well adapted to the natural variability. And we describe these adaptations by something that we call a reaction norm. The very idealized format of a reaction norm is generally a parabolic relationship, this one with temperature, that shows an increase in physiological performance with temperature reaching some maximum level before declining sharply. And an organism's reaction norm is generally sort of created by the interaction between the environmental conditions the organism typically encounters and its genetic makeup. So we can sort of assume or even quantify an organism's sort of thermal plasticity or its tolerance to any other environmental or potential environmental stressor by understanding its reaction norm. This is normally complicated by the fact that multiple traits in an organism respond differently to different environmental stressors. So really, when you're measuring these gross sort of total phenotypic traits, things like survival or growth, they're really the integration of dozens of different reaction norms across several different traits. This is also complicated by the fact that these reaction norms will vary by ontogeny. So as an animal moves from the embryonic to the larval to its adult form, reaction norms are going to shift correspondingly. When we talk about stochastic variability, what we really mean is sharp fluctuations that fall far outside the typical predictability of environmental variability in the habitat. So what I'm showing here is kind of random fluctuations with punctuated and acute moments where the conditions are falling far outside this normal variance. Things like heat waves or cold snaps kind of fit this paradigm quite well. So when we want to understand how an organism is going to respond to these acute stressors, we really have to know what are the most vulnerable periods of this organism's life. And specifically with temperature, it's well studied in marine fish that embryos really constitute this period of acute environmental sensitivity, because normally their environmental tolerance limits, so that reaction norm, are much more narrow than juvenile and adult stages. And in many cases, the ideal temperature can be quite a bit lower than that of the adult. So for those reasons, we believe that fish embryos constitute this real bottleneck of sensitivity. And we know from decades of experimental work and field-based observations that tempered fish embryos show about half the thermal tolerance than their adult forms. And this has been really extensively studied in Atlantic cod by a variety of investigators. And in this species, and probably many other tempered fishes, we know that reproduction is a key sensitivity bottleneck. So we want to understand, if we're going to understand an organism's potential climate sensitivity, we might want to really home in on the reproductive viability with respect to the viability of embryos in early life stages. So as mentioned earlier, I've been predominantly researching warming, acidification, and deoxygenation, but I generally think that these punctuated sort of extreme stressor events represent the most pressing and emergent stressors for marine organisms. And that's not to say that the slow progression of climate change over the course of the coming decades is going to result in wide-scale ecological disruption. But really now and in the coming decades, extreme events are going to be the major driver of a lot of sort of negative externalities happening in the environment. And we already see them. The fallout of the North Pacific heatwave event between 2014 and 2016 were really massive. And only now are we really understanding the ecological toll of that event. There was a huge pool of warm water that spilled across the Eastern Pacific and led to temperature anomalies that persisted for months at a time, resulting in huge effects up and down the food webs, both oceanic and coastal food webs. And effects were felt as far south as Baja California, all the way up into the Gulf of Alaska. So when we think about species who might be vulnerable to these acute events, particularly during the early life stage, I tend to think in terms of forage fish. And that's because forage fishes, as many of you know, are incredibly important to their ecosystems. They serve as a major conduit of energy from secondary consumers up to primary predators, including commercially important marine fish, seabirds, and marine mammals. We also know that forage fishes respond strongly to environmental variability. This is just sort of the nature of their life history traits, in which they constitute a huge amount of biomass. They have huge reproductive events, but small changes in prevailing environmental conditions can really have a huge impact on recruitment success. So year-to-year variability following changes in prevailing climate conditions play an important role in understanding how forage fishes are going to be impacted by future climate change. It's also true that in most systems, while being extremely biomass rich, forage fishes tend to be species poor. So every forage fish species in the habitat is an important target for conservation and management. So understanding how these species, like I said, respond to climate is really a key to understanding the sensitivity of the ecosystem as a whole. Pacific herring are a ubiquitous forage fish on the West Coast, basically all around the North Pacific. And they form these massive spawning aggregations in nearshore environments during winter and early spring, where they adhere eggs in shallow water to submerged vegetation and rocks and other places. But that means that their eggs are in these nearshore environments, so they are acutely vulnerable to both the effects of both marine and atmospheric heat waves. So as you know, we're all familiar with atmospheric heat waves sort of emerging rapidly. These, in some cases, herring embryos fall dry during certain periods of the tidal cycle, so they might be really vulnerable to acute heat stress. But testing this experimentally is hampered by the fact that we don't really know what a heat wave even looks like in a nearshore environment. And what I mean by that are the specific characteristics from the organism's perspective. They're going to influence whether or not a single or individual embryo survives or perishes as a result of a heat wave. So what are the characteristics that are important? Things like the rate of onset. How fast does the heat wave manifest? The maximum intensity, so how hot does it get and how long does it last? These are from the organism's perspective, key characteristics, but from an experimental biologist perspective like myself, we didn't really have an understanding of what these even look like in the nearshore environments to even set up an experiment to test these things. And this is also complicated by the fact that most studies on marine heat waves take the angle of exploring these across sort of huge regional events that occur over the course of months. So not the type of nearshore heat waves that I'm mostly thinking with respect to herring reproduction. So what that meant is that we needed to dig into some observational data to actually understand what a heat wave looked like in a nearshore environment. And fortunately, we have the near site of Padilla Bay as a real treasure trove of observational data. There are several stations in Padilla Bay, which is a coastal estuary positioned in the eastern Salish Sea, located just north of Hidalgo Island. It is mostly a shallow seagrass habitat, and while there isn't a lot of herring reproduction occurring there these days, it is sort of that quintessential herring spawning habitat, shallow, nearshore seagrass beds. And the NEAR system has a few monitoring stations set up there. They've been continuously recording temperature, oxygen, pH variability over the last 2 decades. So this was a really awesome data set to look into to actually explore the effects of heat waves, particularly given that we've had these extreme heat wave events on the West Coast over the last couple of years. And to analyze this data, I use an open source R package called HeatWaveR that has some built-in algorithms to sort of get you started on looking at extreme events and climatological data. So this is a map of the whole Salish Sea, and that red cross there in the top right shows where Padilla Bay is located. And specifically, I used data from the Plogue monitoring station. So this was a monitoring station that basically measured the outflow of the seagrass bed. So the conditions at this station were broadly representative of that over the seagrass itself. So I'll orient each of these figures now. The top left panel is what we call a lolly plot. And each one of these red lines leading up to a small circle is a heat wave event detected in the data set. And the criteria that I used to actually identify heat waves in the data set were temperatures that exceeded the 90th percentile of seasonal climatology for 5 consecutive days. So essentially what that means is over the course of the 20 years, it compared data on every day of every given year, and then looked at where temperatures fell in the 90th percentile on that day. And if temperatures exceeded the 90th percentile for 5 consecutive days, bam, we have a heat wave. So each one of those little lollipops there on that plot shows heat waves occurring in the historical data set. The height of the line represents the maximum intensity. So how hot did things get above the normal climatology for that season? And a couple of interesting things that you might immediately see in this in that time series is that there was a huge cluster of these nearshore heat waves that occurred during the sort of North Pacific blob period. And even though the blob was considered an oceanic heat wave, these data and many other studies now show how the impacts really sort of leached out into these nearshore systems, meaning that there were much more of these high intensity heat events around the blob than there were in previous years. And as I dug into this data a little bit further, generally these heat wave impacts in the systems I looked at correspond to El Nino years. So haven't proven that statistically yet, but it seems like a pretty strong pattern. Anyway, the table on the bottom left shows the top 5 heat wave events with some important characteristics in the Padilla Bay data set. And the plot on the top right is showing a time series of temperature data for one of those events that I picked out. I chose this one. It was the second most intense event in the time series. It occurred in April of 2016. The maximum intensity was about 4.7 degrees above normal. What you see from that time series is that temperatures increased from a normal temperature of about 10 degrees. The solid black line represents the daily average temperature. These individual circles are the individual time or the individual temperature measurements made every 30 minutes during this period. So the average temperature went up from 10 degrees on April 14th, all the way up to about 16 degrees on April 21st. With respect to an organism living in this habitat, that is a huge temperature increase and would constitute serious thermal stress for a lot of organisms in that habitat. And particularly for Pacific herring embryos. Fortunately, there's been decades of research looking at the thermal biology of Pacific herring embryos for obvious reasons. Studies that going back all the way to the 1970s. And with that data, I was able to construct a reaction norm of embryo viability across a range of temperatures. So that's what that black reaction norm is showing. The blue shaded area is the temperatures for which wild spawning occurs in the Salish Sea for wild Pacific herring. Generally from about 8 degrees up to 11, a little bit warmer. And obviously the cherry point stock tends to spawn at temperatures even warmer than that. But that 16 degrees would put viability well outside maximum, and we might expect it to fall by up to half. So if a heat wave would manifest right after our herring spawning event, it could, in theory, reduce the viability of that spawning event by up to half. So this would be constitute a really serious issue because herring, the different stocks, there are 21 of them in the Salish Sea. Each stock tends to spawn over the course of a few weeks. And really a lot of that spawning happens during a few events. So a poorly timed heat wave could really obliterate an entire reproductive output of a stock in a single year. But on top of all that, there's also the emerging threat of ocean acidification in the Salish Sea. Most practitioners of biology and oceanography in the region know that this part of the ocean is what we call a naturally acidified system for a few particular reasons. Some just being the upwelling of deep oceanic water, sort of the retention of water within the Salish Sea basin itself sort of lends to this large seasonal variability in CO2 fluctuations. So this is some time series pulled from Evans et al. 2019, where they looked at CO2 fluctuations amongst other characteristics in the northern Salish Sea. But if you look at this middle plot here in C, you see that during the winter, CO2 levels can exceed 800 microatmospheres. So that's double atmosphere. So really, these punctuated acidification events theoretically could pose an additional stressor on top of things like heat waves. What this means for the organisms living in this habitat are important. First, it means that Salish Sea ectotherms must be adapted to these natural CO2 fluctuations. There's no way around it. They experience these as a natural state. And as a consequence, they must be able to tolerate these natural fluctuations. But that also might mean that they're living much close to their physiological tolerance limits. So even though they can withstand contemporary fluctuations, even a small increase due to the anthropogenic signal might push them over the edge. Sort of like a tropical species living on the equator, like there isn't a lot of room for further increases. So understanding the combined effects of these thermal stress on top of acidification, while complicated, is really important for predicting tolerance or really resilience of these important organisms. So one of the key hypotheses in ocean acidification research are looking for these interactive effects. And one of the key questions is, does ocean acidification exacerbate heat stress? And the underlying physiological rationale for this is that elevated CO2 in the water increases the metabolic demand of acid-base regulation in water-breathing animals. That is, animals have to work harder, they have to expend more energy to maintain acid-base homeostasis in their tissues. For fish embryos that have a very limited ability to partition energy supply to different homeostatic mechanisms, this might push them over the edge in terms of their energy supply limits. So thermal acclimation in ectotherms generally requires an increase in metabolic rate as temperatures increase. This is, classic thermal biology. But as temperatures increase, if there's these added stressors being mounted on top of that, it might exceed the amount of energy available for rapid acclimation, resulting in death or other malformations. So this is why we think, or why a lot of people studying ocean acidification, even though CO2 on its own might not represent an acute stressor, in combination with some of these other stressors, it might reduce the resiliency of an organism. And this is why we have to study it. So this is just a theoretical reaction norm of what a typical exposure to the simultaneous acidification might do to an organism under thermal stress. So a couple of key hypotheses that we went into this study. The first is we predict, given on the established reaction norm or thermal reaction norm of Pacific herring embryos, that under the heat wave like I just described, we would predict to see a decrease in survival. And that combined with high CO2, this will aggravate the physiological effects by increasing energetic demands associated with acid-base regulation, while simultaneously constraining the energy supply that's required for rapid thermal adaptation. So these experiments were conducted at Shannon Point Marine Center, part of Western Washington University on Fidalgo Island. It's a campus that I miss dearly now, living on the dreary East Coast, not able to look behind the lab and see this every day. So here's the experimental design. I conduct these experiments at Shannon Point's wet lab space. And what I did is I constructed these 16 individual flow-through units, which I was able to manipulate the temperature and the CO2 level in each one independently. So I had these 16 independent units. The water flowed from the header tanks into these small buckets where the individual herring embryos were being housed. These embryos were taken from wild spawners that were collected from Port Gamble in late February. I dissected the gonads, I brought them back to Shannon Point, and then performed the manual fertilization using some established protocols. Once I confirmed fertilization success, I then split these embryos up into 4 different treatment groups. So I had ambient temperature, which was just the temperature of the flow through coming into the lab, which is about 9 degrees during the course of the experiment. And I cross that with two CO2 levels. Control, about 550 microatmospheres, which is about what we were seeing in the surface water during the course of the experiment. And then high CO2, about 2300 microatmospheres, which is more than 5 times contemporary atmospheric CO2. But it does represent sort of the upper limit of acidification in this system by the end of the century. So given various predictive models under various climatological scenarios, we could see at least seasonally levels that high by the end of the century. So it's also an important benchmark for a lot of acidification research because of that. If organisms can tolerate levels as high, then it's unlikely that ocean acidification is going to be a stressor for that particular species. I then crossed those two CO2 levels with 2 heat wave treatments. So the heat wave treatments are a little bit complicated, but the schematic down below will give a graphical illustration of how this went down. I let all the embryos develop for 3 days under that sort of ambient temperature regime. And then day 3.5, I started the temperature ramp up. And I designed this temperature ramp up sort of with those most intense heat waves I found in the Padilla data set. So over the course of 5 days, I increased the temperature by about 0.85 degrees a day to result in about a 4.5 degree increase. So this essentially mirrored that really intense heat wave that we saw in April 2016. So by day 9, the embryos were now being exposed to about 13.5 degrees relative to the control group. So during the onset of the heat wave, and then once these embryos hatched, I measured a couple of key response traits. One is that I measured routine metabolic rate via closed oxygen consumption measurements during the onset of the heat wave to get an understanding of how these different stressors were affecting the metabolic rate of embryos. I paired that with yolk consumption measurements to get a more integrative look at energy consumption across the entire experimental period. So oxygen consumption measurements give me this sort of instantaneous look at physiology. Yolk consumption gave that integrated over the course of the exposure. I also looked at 8 different morphometric measurements of newly hatched larvae. And along with that, I was able to identify some gross developmental malformations that ended up being pretty important. And then finally, just standard hatching success or embryo survival. So the data I'm going to show first are from the metabolic rate data. So to orient you here, on the y-axis is routine metabolic rate in nanomoles of oxygen consumed per individual embryo. And then this is all standardized to how large the embryo was. And then on the day or on the x-axis are the 3 days that I measured these respiration rates. So day 6, 7, and 8 post-fertilization. And this little inset here is showing the actual temperatures during the assays. So the bottom are the control temperatures are always at 9 degrees. And then the top line shows the increase in temperature over the course of the measurement period. So what we see predictably is that the fish from the heat wave trial were showing significantly higher respiration rates. And this was no surprise. Fish have higher metabolic rates when temperature is warmer, combined with the fact that these embryos, having been exposed to warmer temperatures, were now at a later developmental stage versus those at the control. So this increase in metabolic rate is a combination of temperature and developmental stage. But the most important thing to take away from this figure is that I don't see any significant effect of CO2 on respiration rates at all. If anything, there was a slight decrease in respiration rate under high CO2 at the ambient temperature group. So our hypothesis that CO2 increases standard metabolic rate and in conjunction with warming temperatures was nullified. I found no evidence to support this hypothesis. If we look at the different morphometric traits of newly hatched larvae, again, we see that temperature has the overwhelming effect. Fish from the heat wave treatment were hatching significantly smaller size, more than 0.5 millimeter than those reared under the ambient temperatures. This is a pretty conserved trait across temperate fish, and that warm conditions generally produce smaller larvae. So this is what we ended up seeing here. But it kind of varied by the specific trait. So in terms of standard length and somatic body area, it turned out the same, but some other key traits, we didn't see the same response, particularly with respect to dry weight and yolk sac area, which I'll get to in a little bit. So then I took those different morphometric traits and developmental rates and computed some key rate data. So the top panel here is showing developmental rate. So this is essentially time to hatch, and it's shown as a % developed per day. The middle panel is showing yolk consumption rate. So again, showing how much yolk as a function of % that these organisms were consuming under the different temperature and CO2 levels. And the third is growth rate. So it's simply how many millimeters did these embryos grow from fertilization until hatch as a function of days. And what you'll see here is, again, that temperature had the overwhelming effect. Fish from the heat wave, or the embryos from the heat wave trial, grew faster, they consume their yolk faster, or they develop faster, consume yolk faster, and grew faster than their conspecifics under the ambient. Again, no surprise. But the important note here is that CO2 had no significant effect on any three of these traits. It was completely temperature driven. So what I did from there is I wanted to compute Q10 ratios for each one of these rates. And the Q10 is a really sort of vital metric because it's able to allow you to compare different rates when they're measured on different units and maybe computed across different temperature ranges. Because one of the complicating factors of this experiment is that I was evaluating these different rates during different time frames. So actually the specific temperatures that were being manipulated over the different time frames were slightly different. So they're not actually comparable one-to-one. But standardizing them by Q10 allows me to get a much more clear look at what the effect of temperature was on these different rates. And just a quick understanding of what the Q10 equation looks like. It simply standardizes any change in rate to a 10-degree temperature change. So even though I only changed the temperatures in this experiment by 4.5 degrees, all these rates are now standardized to a 10-degree temperature change. So on the left panel, I'm showing the developmental rate and yolk consumption rate. They each had a Q10 about 3, which is very consistent for tempered fish embryos with respect to these two traits. So in different review studies I looked at, a Q10 to 3 is very common. However, growth rate showed a much lower Q10 of only about 1.8. So there was this disequilibrium between the three sort of key rates I measured on development, yolk, and growth. The literature suggests that all these three should respond in unison. What I found is that growth rate had a much lower Q10. And what that means is that the production efficiency of these embryos dramatically fell when they were exposed to a heat wave. And what I mean by production efficiency, if you think of it this way, it's simply the amount of yolk that these fish consume, because remember, embryos have a fixed energy supply given to them by their mothers, and they essentially convert that yolk into body mass. So if you're able to describe this ratio of the amount of tissue produced versus how much yolk consumed, you're able to get this production efficiency metric. And what I found is that fish under the heat wave treatment showed a 10% reduction in production efficiency, which doesn't sound like a lot, but size is everything when it comes to the survivability of these early larvae with respect to predation, starvation resistance, and hunting success. So the fact that they're ending up with this 10% reduction production efficiency could be the difference between having a boom year and a bust year. But again, one of the key findings is that I found no evidence that CO2 affected production efficiency on top of the heat wave. Another surprise from the study was that the heat wave actually increased total hatch, not by a significant amount, by a little bit. So under the ambient temperatures, total hatching was about 80%, which is pretty normal for this range of temperature. But we actually saw an increase, which was very surprising because the previous studies that looked at static exposures to different temperatures suggested that we should see a decline in hatching success. But what we found using this heat wave approach was actually a positive effect in hatching, which was completely opposite of what we hypothesized. And this is probably because embryos actually increase their thermal plasticity as they develop. We'll talk about this in a little bit, but it was a really interesting finding. But what we also found is that the heat wave significantly increased the proportion of malformed larvae. So even though more were hatching, there was a higher percentage of these were coming out malformed. So kind of equalized the ultimate survival of the embryos, because these malformed larvae, while they had a beating heart, were not actually going to survive in the water column. So in the end, hatching success was more or less equal across the 4 groups. A couple of examples of malformations like this hook here, which is pretty characteristic, but stunting, really long, narrow bodies with edema around the heart and yolk sac, and these really dramatic sort of hooks in the spinal column. So in the end, what we found was this, despite predicting about a 20 percentage point decline in survival, we didn't see that at all under the heat wave. So all in all, what we found is that despite there being some important bioenergetic effects, these embryos showed a remarkable tolerance to the combined effects of what would be a pretty extreme heat wave, combined with very high CO2. So this is good news for Pacific herring embryos. They show a lot more environmental plasticity than I predicted going into this trial. But a couple things that demanded immediate follow-up. The first is that we evaluated the heat wave period across embryonic development. So the heat wave was initiated right after the closure of the blastula, which is typically a very sensitive stage of environmental tolerance in fish embryos. But we didn't reach maximum heat wave until the eye stage. And by the time an embryo reaches an eye stage, its thermal tolerance is actually quite similar to that of the larvae. So we moved from what is a very sensitive to a somewhat tolerant stage. So maybe we need to focus a little bit more on these early embryos if we wanted to identify potentially issues with sensitivity. The second thing is that if you look at the sort of high frequency fluctuations during a heat wave, I mostly designed this heat wave to follow the average temperatures that were occurring in each day. But in reality, because this is a near shore tidal environment, you have these huge swings in daily temperature. And in fact, the maximum temperatures that an embryo would have seen during this heat wave would have been close to 20 degrees. So much higher than the 13.5 that we peaked out in our study. So this demanded that we design an experiment a little bit differently to look at these more acute effects. So I worked with some folks at Western Washington University, namely Brooke Love and her master's student at the time, Nicole Singh, to look at the different effects of different spawning groups on these acute heat wave effects combined with ocean acidification. And when I think about critical windows, I know that I was describing it in terms of like the whole embryo stage earlier, but this point just at gastrulation. So when that initial sort of shield of cells grows around the yolk, that tends to be the stage at which embryos show the narrowest limits of environmental tolerance. And this has been showed really conclusively in Atlantic cod. And again, during our period, I only evaluated respiration rates with respect to acidification and heat wave effects in embryos that were about 50% developed to about 70% developed. So we completely missed out on these really early stages. So in this follow-up study, I wanted to sort of hone in early developmental stages to see if we see any differential patterns. And indeed, I found something really interesting. So I took embryos that were being reared under chronic temperatures of 10, 12, 14, and 16 degrees and 2 CO2 levels. And then using the same exact methods as I did in the previous study, I evaluated their respiration rates. And this time, what I found was about a 30% increase in respiration rate under high CO2. So this was completely flipped from what I had found previously. And what it suggests is that developmental stage indeed will predict the response of embryos, metabolic response in embryos with respect to acidification. What I should caution is that even though we see this significant increase here, on the whole, I don't think that acidification has much of a metabolic effect on these embryos. So despite the fact that it increases metabolic rate in one stage, if you integrate this effect across all developmental stages, the effect is still pretty small. But if we're going to be looking at acute effects, then this embryonic stage probably is where we should look, given these early results on respiration rate. So what we did is designed a much more complicated assay. And we took embryos that were being reared under chronic 10 or chronic 16, and then exposed them to this acute stressor assay. So we took embryos, we put them into these incubation vials, and then we warmed them up by 1 degree every 15 minutes until we reached either 20 degrees or 25 degrees. So very warm for these embryos. And then at each one of those maximum temperatures, we held them at either 1, 2, or 3 hours. So we essentially formed these 6 different experimental treatment groups: 20 and 25, 1, 2, and 3 hours. And this sort of mimics how embryos will be exposed to acute temperature stress in the environment. So you imagine a really extreme nearshore heat wave. As the sun sets, temperatures might drop. As the tide rises, temperatures might drop. But still, embryos might be exposed to these acute temperatures for about an hour, maybe 2, maybe 3. So that's what we were looking to get at. But what we found was really some surprises. We didn't see any significant decline in embryo mortality as measured 2 days after the initial exposures at any of the 20-degree exposures. So despite being held at 20 degrees for 3 hours, most of these embryos were still alive as compared to the control group 2 days later. Well, we did start to see some mortalities in the 25C, particularly at 3 hours. So once you start holding these fish or these eggs for 25 degrees, they're no longer viable anymore. I saw a lot of gross embryonic malformations and a lot of embryos being arrested in development immediately during the assay. So they succumbed to the heat stress during the assay itself. But again, from here, the overall effect, I'm not showing it in this figure, but there was no effect on CO2 and any survival parameters. So despite the fact that we saw that increase in metabolic rate during this developmental stage, even though we expose them to this acute heat stress, still no significant effect of CO2. So if I have just a couple minutes here, I'm going to quickly run through another study I did in collaboration with the Western Fish Health Science Center and Marrowstone Labs. And I think it's really important because it's focusing on the long-term effects of acidification and interactions with disease. And this is an emergent topic that I think is key, but remains acutely understudied with respect to fish. People who study climate change, generally have this worry because there's been a spike of marine disease outbreaks in a number of groups. And often this is associated with acute stressors like heat waves. This has been seen in corals and a number of other invertebrates. But with respect to acidification, the science is much less well established. We know that chronic exposure to acidification alters immune physiology in a number of calcifying organisms, namely commercially important shellfish, Pacific oyster and blue mussel. In some cases, the acute exposure to acidification hardens them against disease. In some cases, it makes them more vulnerable. There's also sort of long-standing evidence from aquaculture scenarios that really acidified, very high levels of CO2 in aquaculture settings leads to outbreak disease. But that's more observational than being proved experimentally. So what I want to do is work with the Marrowstone, USGS Marrowstone Marine Field Station, where they do a lot of work focused on disease ecology of Pacific herring. They really focus on viral hemorrhagic septicemia virus. It's an endemic virus in temperate fish across the Northern Hemisphere. It's been implicated in a number of fish kills in Pacific herring and from Alaska throughout the Canadian and the West Coast of the United States. There's a lot of work done by Paul Hirschberger and his crew at Marrowstone, sort of describing the ecology of these outbreaks. But there's still a lot of unknowns about what sort of is the mediating driver of these events. So the main focus of this study was that we reared herring under 3 levels of CO2 from hatch until a late juvenile stage, until they reached about 5 to 6 centimeters in total length. We reared them under 500 microatmospheres, again, ambient for the surface waters of the Puget Sound, and then 2 elevated levels, 1500 and then 3000, which is a very high level stressor that probably doesn't exist in nature in the Puget Sound. Along with the disease study, we wanted to measure growth, and this long-term study presented a really good opportunity to see what the long-term effects of acidification are on development. This is data that is really sparsely investigated with fish, mostly because it's hard to do experiments on fish and acidification in the long term. So this was a really good opportunity for me to sample these tanks at high frequency, and if there was a growth effect of CO2, I would find it. What these figures are showing you is essentially there was only a very small effect on growth at only the highest CO2 level. So over the course of about 100 days of experimentation, there was only one tank under high CO2 that showed a significant divergence from any of the others. Two of the other replicate tanks showed identical growth rates as the control groups. So even though you see a small decline in growth rate with that red solid line with respect to standard length, that is mostly a tank effect. And there was no difference between the relationship of dry weight and standard length, which is important, essentially, which is the condition of the fish. So CO2 had no effect on the condition of the fish. So just looking at these plots, we would assume that there wasn't really a chronic stressor being imposed on these organisms, despite the chronic nature of the high CO2 exposure. Also, I measured critical swim speed in these late-stage juveniles. Burst swimming is a really interesting trait to measure with respect to chronic stress, because just like sprinting and as humans and sprinting or marathon running, it is integrating a lot of physiological functions in a way in which an unhealthy organism is going to show significantly lower ability to burst swim versus a healthy functional organism. So I swam these guys in a swim flume, and I used a slow ramping methodology to figure out what their sort of highest sustained swimming speed can be. And I measured 15 individuals from each one of the CO2 groups. On average, the fish from the control group had about a 10% higher critical swim speed versus those from intermediate and high CO2. However, the variances were huge. And this essentially rendered any statistical significance to the data set a moot point. I think this mostly speaks to the fact that swimming herring and swim flumes is very challenging. And even though I swam 45 fish for the study, I had to practice on dozens before I got a good methodology down. So despite not finding a significant effect here, I think maybe with some refinement of methods, we might be able to look for more subtle effects and the chronic effects of acidification in juvenile herring. So here's just a quick video of what the little herring in the swim flume looks like. And this guy's swimming about as hard as a little juvenile herring can. So onto the disease study. We used Maristone's quarantine labs to set up 18 individual tanks, 6 per CO2 level. After 2 days in the tanks, we inoculated half of them at each CO2 level to VHS. And we tracked mortality over the course of 4 weeks. So the big figure on the right here is showing the mortality curves under the actually 6 different treatments. So 3 CO2 levels and then 2 disease exposures. The top line is showing the control tanks, essentially no mortality in these ones. But then the lines shown with the triangles, those are the disease exposed colonies. And they show a really classic decline in survival that starts about 5 days post exposure to VHS. Fish become infected with disease. Those that are really susceptible become super spreaders in the tank. This will then sequentially affect other fish, and then the disease basically takes off from there. What we found was pretty interesting though. Immediately, there was a huge decline in survival in the fish from high CO2. So by day 6, it had dropped to 70% survival relative to 85% under the control intermediate CO2. Or over the course of the weeks, there was basically a leveling off of mortality under the high CO2 line, whereas mortality sort of continued to pace under the control tanks. By the end of the experiment, there was, on average, higher mortality in that high CO2 tank, but statistically not different than the other 2 treatments. We also looked at the prevalence of the virus in tissues of both fish that died during the course of the experiment and those that survived to the end. And in essence, there was no significant difference between the amount of disease that we were finding across CO2 treatments and that of the survivors. So any fish that survived to the end of the treatment basically had no detectable amounts of the virus in their tissues. So while there was some initial evidence to suggest that high CO2 fish were maybe more sensitive to that initial burst of disease activity. And at the end of the day, it doesn't appear like CO2 plays a significant role in viral resistance of Pacific herring. So with that, I'd like to acknowledge particularly the Washington Ocean Acidification Center for funding all this research. Terry Klinger and Jan Newton, fantastic supervisors during my time at UW. The folks at Shannon Point for letting me sort of station there and set up shop for a lot of experiments. WDFW for helping me get wild fish samples every time I requested. And obviously the folks at USGS Marrowstone for having me 2 summers ago. It was probably the best research summer of my life. With that, I'll be happy to take any questions.
Wonderful. Thank you so much, Chris, for your presentation. And I'm sitting here and my mind is swirling. Do we have any questions from folks? You can either put it in the chat, or if you want to raise your hand and let's ask Chris directly, you can. So any questions from anybody in the audience today? And if you can't figure out how to raise your hand, if you just want to unmute yourself, we can do that as well. Okay, so I don't see any right now, so maybe people are a little shy, but you know, I was thinking about, I've read the paper obviously, and that's how I was able to find you and to do this seminar today. And I think one of the things that this really brought home to me was, I think herring are a lucky species because they spawn in April, you know, in that winterish, you know, spring time period. What do you think would have happened if they would have been spawning during our incredible heat wave in June of 2021? The last one where we had beach temperatures over 100 degrees. Or what would happen to the developing larvae, do you think, or the small fishes that are out in the estuaries at that point? Would they have been okay?
Probably not.
Okay.
I think one reason why I like to focus on embryos is because they're sedentary, right? They can't evade these warm pools of water. To some extent, even larvae, depending on the species, are more or less sedentary, who are just sort of going with the flow, more or less. I think adult fishes tend to seek out cooler water in these events, but that's going to have a lot of knock-on effects on it in and of itself. So I think that fish larvae, embryos living in shallow nearshore environments during extreme heat stress are extremely vulnerable, either through direct impacts of the heat or oxygen limitation, because often heat is associated, particularly at night, with low O2 and high CO2. So these combined effects would certainly produce a lot of die-off. But some herring, Pacific herring, do spawn in the Salish Sea during June. Almost exclusively the cherry.
Absolutely.
So the cherry point stock, I think they show this separation in spawning phenology. But at least our initial work, I didn't show it yet today, a lot of it today, but we did some comparative analysis between the cherry point herring and those that spawn earlier. They didn't show an increased tolerance to temperature in any way. So despite spawning later in the year, I don't necessarily think they're more well adapted to extreme temperature stress. I think that spawning phenology in herring generally is a plastic trait that tends to shift across the millennia or the centuries, depending on prevailing conditions. And if you go to different herring across different latitudes, they might be doing things much differently than the Salish Sea. As a consequence, they sort of retained a lot of plasticity, which allows them to tolerate a wide variety of environments. But there's definitely an upper thermal limit to their ability to withstand stress.
Do you, and one last question, and then Jenny had her hand up. Do you think that, you know, we're in the lunar nodal cycle right now, the 18.6 year cycle, where we have extreme lows at key times, at least in the shellfish world, but also I imagine in the herring world as well. And luckily we generally don't see a lot of sun or a lot of heat, you know, like we do in the summer in April. But what would that do to the stock?
Yeah, sort of embryos falling dry is a really interesting phenomenon. They're well equipped to do so. Other species in the Salish Sea even spawn higher in the intertidal than them. I'm thinking sand lance, So falling dry in heat stress and sunlight, yeah, it could be a big problem. On the other hand, once the eggs are in the atmosphere, they're no longer limited by the oxygen limitations of the aquatic environment.
Mhm.
So being exposed to air during extreme heat stress might actually, especially if they're sort of covered in macroalgae, might be beneficial in some ways. And
Okay.
honestly, is very situational dependent.
Mm.
Sunlight too, but one thing I would say that at least climate predictions for the Pacific Northwest suggest that anomalous heat events are going to occur earlier and earlier in the year. So while we're very used to these very mild cloud covered springs, that might change over the coming decades.
Mhm. Wow. Jenny, you had your hand up. Are you still interested in asking a question?
Thanks, Terry. Yeah, Chris, great presentation. Thank you so much. I actually have kind of two questions. One is, would you expect to see these same conditions or same effects on the coast, on the outer coast of Washington? Or do you think it's specific to the Salish Sea? And then the other question is really about other forage fish that you may have the same expectation about other species. You mentioned sand lance, for example. I know they have a different life history, but what other forage fish do you think might behave in the same way? Thank you.
Yeah. Um, the herring on the coast are interesting. They have a very different life history cycle than the more resin imputed soundfish. So a comparative study would be really, really interesting. I don't know about their spawning phenology as much as the Salish sea stock. So I don't know, whether or not the timing of their spawning runs might influence their potential sensitivity. One thing I do know is that herring are, there's a lot of mixing that occurs between stocks, except for the cherry point stock. So even though we isolate them into these 21 different stocks, most of them are genetically homogeneous. So I would expect these responses to be pretty conserved amongst the winter spawning, spring spawning Pacific herring up and down the West Coast. I think maybe really big latitudinal differences going from Alaska to Northern California, you probably see some thermal or some variation in the thermal biology. And that's obviously been demonstrated in a lot of species. But even so, the gradient, temperature gradient is not huge between that distance. So I do think that herring generally show a pretty wide plasticity with respect to thermal tolerance. With respect to other forage fishes, this is really interesting because I've been working on a species of sand lance in the North Atlantic since 2014 or 15. For the rationale that it's a species that spawns during winter in the offshore where CO2 fluctuations are very small. And what we found is that high CO2 inhibits the ability of the sand lance to hatch, at least in the laboratory. And the effect is very dramatic. It reduces hatching success by more than half in many of our experiments. At levels of CO2 only about 12,000 microatmosphere. So we're working on actually identifying the mechanism involved in this CO2 impaired hatching hypothesis. And one thing I really wanted to do when I was in Washington was test this hypothesis on Pacific sand lance. Because Pacific sand lance that are living in the Salish Sea, again, are in this naturally acidified system. They're spawning during winter when CO2 is at its seasonal maximum. So if there's adaptations to be had, this would be the species that would show it. So this sort of comparative within genus study of Pacific versus Atlantic sand lances, I think would be really powerful for showing the ability for localized adaptation to CO2 fluctuations.
So that
Thank
feeds
you.
right into Liz's comment and a question. Herring spawn much earlier than April in some Oregon estuaries, and she wants to know if you have observed greater stochasticity in spawning time by weeks or months, or shifts in timing in Washington or elsewhere in the Pacific Northwest.
Yeah, so the Washington Department of Fish and Wildlife has an excellent resource for spawning phenology of Salish sea herring. So I'd really encourage you to check out I think they have a recent stock assessment that was published this year or last year that well documents the spawning phenology of all the stocks. There is and has been some movement with respect to spawning phenology for many of the stocks. I don't know if it's clear whether or not this is just sort of the natural way of things, or if this is a true response to temperature driven effects. I would say that for the most part, Contemporary warming in the sales sheet hasn't gotten to the point where it's probably affecting this phenology too much, not yet.
So Paul Hirschberger had his hand up, and then after him, Phil Dion. Paul, do you still have your question?
Yeah. Thanks, Chris. That was a great talk. I enjoyed it.
Thanks, Paul.
I have to admit, I tuned in a little late, so you may have answered my question. I think a lot of these phenotypic metrics that you're looking at are quite variable, in terms of Euclid and the metabolic metrics and the developmental metrics as well. I wondered if you had looked into any sort of transcriptomic effects with PCO2 levels and temperature.
Yes, so this is the focus of my current postdoc now at Woods Hole. The problem with transcriptomics is obviously it takes forever to develop any sort of real results. So don't have too much of a question, or don't have an answer with respect to herring and that front. What I will say is that for most studies that have focused on things like RNA-seq, looking at effects of CO2 on fish embryos, most of the time CO2 results in a very broad changes in transcriptional responses that you would expect to see very pronounced phenotypic change where you don't actually see the change. And that's gotten a lot of investigators in the field to sort of declare that CO2 acts as a stealth stressor, and that we don't see, like you said, we have a lot of variety in these morphometric traits or other sort of metabolic assays. But on the average, we don't have a lot of difference between different groups. But then you look at the transcriptomics and you see these enormous responses in almost all the fish you study, a complete overhaul in different pathways. So it does suggest that CO2 acts as a stressor in the environment, but fish, particularly that live in variable environments, have these built-in mechanisms to sort of reshuffle their physiology in a way that sort of reduces the overall impact. Even so, even if we don't see these punctuated responses, a reshuffling of your metabolic programming might have knock-on effects across life stages. So again, we focus on herring in our study, and we want to know whether or not there's these carryover effects of continuous exposure across juvenile development. For the most part, it wasn't there, but we see a lot of variability. There are probably some genotypes of herring that just don't respond very well to high CO2 for whatever reason. Perhaps we can get at a more mechanistic understanding if we're able to do some more of this transcriptomic work.
Great. And Phil.
Thanks. I just wanted to follow up on Liz's question. We have looked at the spawn timing in Puget Sound for herring, and our spawning can be as early as December, and like Chris said, goes all the way through June some years, depending on which stock we're looking at. And we have seen shifts, but when we looked at that, we didn't see anything consistent. We had some stock spawning earlier and later, so there doesn't appear to be any relationship with the changing water temperatures. And also to Chris, great talk. If you're interested in looking at those sand lance in the Pacific Northwest a little bit more closely, get in touch. We might have some means of getting hands-on fish for you.
Yes, getting hands-on fish is really the key here. The experiment is very easy.
Well, great. Thank you, Phil, for that wonderful offer. And we have another question in the chat relative to the previous question. In their limited knowledge, transcriptome changes are also highly variable in response to partial carbon dioxide. They're just curious what you think about it.
Thanks, Neil. Again, I think what this essentially means is that like most responses of fish early life stages, is that there's a huge amount of variety because fish early life stages tend to be much more genetically diverse than their adult stocks, right? Because there's a huge selective force acting on fish early life. People cite the overwhelming amount of 99% of fish larvae succumb ultimately prior to recruitment to the fishery. So this huge variability we see in responses is a function of the fact that the genetic pool of early life stage fish in which we're measuring is much more diverse. The key here is to understand who are the winners and losers, of these stressor effects, particularly under multi-stressor environments, and then understanding what type of phenotypes is produced in the long run. So even though we don't see a lot of effects in terms of survival and growth of these earlier life stages, is there a selective force being acted upon these fish that's going to further narrow the genetic diversity at a later life stage, which could have knock-on effects for stock viability, growth rates, maturation timing, lifetime reproductive output. These are all things that are extremely difficult to measure in the lab, for most fish at least. So designing experiments in which we can look at different pieces of this using a lot of different tools, integrating them is how we eventually get to that answer.
Great.
Yeah, no, thank you. I was just curious more in terms of what's the ultimate goal, by understanding the effects of PCO2 and heat waves and these combinations of stresses, like what is the ultimate goal that managers or regulators need that we can provide in terms of conducting physiological or molecular endpoint, understanding more about molecular endpoints? So
I think the first is that this type of experimental work will establish like limits of resilience. So one thing managers can do is sort of advise on like local pollution, right? If we're worried about coastal acidification and hypoxia, if there are things that managers can do to mitigate those levels of stress in the near shore, if we know, sorry, the cat's yelling at me for dinner. If we know what those levels, those tipping points be, we might be able to avoid them. Transcriptomics, I feel like, is a little bit more far removed from that. Like we get an answer, we can then inform management. Because oftentimes people don't pair transcriptomic responses with like really easy to interpret physiological assays. It's just too much work. I think what transcriptomics provides is different pathways of sensitivity. We can identify what are the actual mechanisms of stress that are underlying our whole animal physiological response. They can inform us about different experiments we can take to measure these different traits a little bit more fine-tuned. And I think that it also allows us to maybe recognize things that we just can't see in our whole animal physiological assays. I think there's a really good paper on Alana cod that demonstrates this, that they see a lot of variance in mortality of cod that can't get a specific mechanism of high CO2 sensitivity. What they see in the transcriptomic data is a complete reshuffle in a variety of physiological pathways. And using that information, they were able to establish some new, more targeted assays in terms of the physiological responses to figure out what's happening to these fish.
thank you.
Yeah.
Wonderful. So this brings us to the end of our session today, and a video recording of this session will be available on the California Current Acidification Network website shortly. You can join the CCAN listserv at that time under that same, and you can see future webinars. You should also check out the Ocean Acidification Information Exchange and join the CCAN section. And I want to thank you and Chris especially for presenting today, for participating, and we welcome any feedback. Further questions, I've put Chris's email in the chat, any suggestions for future topics. And thanks again, Chris, for being here, coming out to the West Coast virtually for this today and sharing this. And thank you everyone for participating. 1 second.